Protein synthesis begins with transcription inside the nucleus. The enzyme RNA polymerase binds to the promoter region at the Tau box and begins unwinding the DNA. In our model, the top strand serves as the 3' to 5' template strand. Now, RNA polymerase moves along the template, reading the DNA bases to build a complementary mRNA strand to the 5' to 3' direction. Note that in RNA, uracil replaces thymine to pair with adenine. The polymerase also assembles the sugar phosphate backbone as it goes. The process continues until the RNA polymerase reaches a terminator sequence. At this point, the enzyme detaches and the newly formed pre-mRNA is released. As the polymerase leaves, the hydrogen bonds between the template and coding strands of the DNA reform, zipping the gene back together into its original double helix shape. Before leaving the nucleus, the pre-mRNA is modified to become mature mRNA. A 5' methylated cap is added to the front to help the ribosome recognize the strand. At the 3' end, a poly A tail is added to protect the mRNA from breaking down in the cytoplasm. Finally, non-coding introns are removed, leaving only the coding exons. Now, translation. It begins in the cytoplasm. The small ribosomal subunit binds to the mRNA and scans for the start codon AUG. An initiator tRNA carrying the amino acid methionine matches its anticodon to the start codon. Finally, the large ribosomal subunit joins to complete the initiation complex. Now elongation begins. The ribosome has 3 functional sites, A, P, and E. First, a new tRNA matching the next codon enters the A site. Here, it's carrying proline. Next, a peptide bond forms. The growing polypeptide chain is transferred from the tRNA in the P site to the amino acid on the tRNA in the A site. Then the ribosome translocates or shifts 3 nucleotides toward the 3' end. The empty tRNA moves to the E site and exits. This cycle repeats, growing the protein chain 1 amino acid at a time. Elongation ends when a stop codon enters the A site and termination begins. Since there is no matching tRNA, a release factor protein binds instead. This triggers the release of the completed polypeptide chain and the disassembly of the ribosome. Protein synthesis is now complete.